So this brings us to the next main topic, and that is the enzyme. So because metabolism is controlled by enzyme, 2-3 times I have spoken this statement in the last video which have you seen. So what are enzymes now? So we can also call enzymes as the biocatalyst. Now you know in chemistry what is catalyst, something which increases or something which decreases, or you can say something which alters the rate of reaction and is always required in a very small amount, or you can say traces, and they are not consumed in the reaction. So at the end of the reaction they will be as such present there, or they will not even undergo any change. Such molecules are called as catalysts. And remember, they can increase the reaction, so they can have a positive effect on the reaction, or they may have a negative effect on the reaction. So such molecules, when present in the living system, are called as the biocatalyst. So please remember these main features, that enzymes are biocatalysts, and they change the rate of reaction. Then they are required in a very small amount to control the reaction, and they are not consumed or they are not changed during the process of reaction, and they may have a positive effect on the reaction. If they increase the rate of reaction, it is positive. If they decrease the rate of reaction, they are called as having negative effect. And then mostly enzymes are proteinaceous substances, except a few which are made up of RNA. This topic I have already covered in the types of RNA, if you remember, catalytic RNA. So again I have written some enzymes here. These enzymes are made up of RNA. Example: ribozyme, ribonuclease P, peptidyl transferase enzyme, telomerase enzyme. Remember, these are all in your 12th class syllabus, not now, because you learn about the complete genetics in the 12th class. So these topics will come then. So and one more thing, enzymes will have active sites. If you have seen the videos carefully, in the protein topic I kept telling you that they form the active sites of the enzyme. So tertiary structure plays a very important role in enzymes, and remember, enzymes are globular in nature, globular in shape. Now the next thing, who discovered enzymes for the first time? So the catalysts were discovered by Berzelius. The name I have mentioned here. He discovered the catalyst for the first time in 1853, and they are involved in catalysis. Catalysis is the phenomena of change in the rate of reaction in the presence of small amount of specific substances called as catalyst. And then the next point main to remember is that enzymes were, so who coined the word enzyme? So it is Willy Kuhne in 1878. He used the enzyme word for the first time, but who popularized this name? It was Buckner in 1897, who is credited with the actual discovery of enzyme. Now, why Buckner is so important? Because he was performing experiment on sugar and yeast. We all know when sugar solution when comes in contact with the yeast, then slowly the yeast will cause fermentation of sugar, and finally the sugar solution will change into alcohol. So this is done with the help of living yeast. Now what he did, he crushed the yeast and then created an extract out of yeast. So that means a dead thing. A dead thing is there because yeast has been killed, and he took the chemicals or the solution or extract out of the crushed yeast, and he called that extract as the zymase, and he added that to the sugar solution. So now it is not the living yeast but only the extract of the yeast. And he found in the same manner like yeast perform fermentation, even zymase is able to cause fermentation of sugar solution, and the sugar solution changed into fermented sugar, and finally alcohol was obtained. So what he found in this whole process was that it was not the living system which was able to convert sugar into alcohol. It was the extract of the living system, zymase, which he called, which was able to change the sugar into alcohol just like the yeast does. Therefore, first enzyme was isolated by Buckner in 1903. And then remember, Sumner in 1928 purified and crystallized the protein for the first time. So Sumner purified them. He only isolated, purification was done by him, crystallization by him, and then which enzyme he crystallized is the urease enzyme. Remember, Sumner also stated that the enzymes are proteinaceous in nature. He also stated same point, but this was confirmed by experiments with the experiments performed by the Northrop. So Northrop even received Nobel Prize for this, because he confirmed in 1936 that the proteins, the enzymes are proteinaceous in nature. For example, pepsin and trypsin. Now remember, some enzymes can be made up of RNA. So the first RNA enzyme was ribozyme, and it was obtained from ciliates. So protozoa which carries ciliates, and the name of the organism is Tetrahymena thermophila in 1981. And remember, this enzyme, also called as the ribozyme, is a self-splicing intron. Now what is splicing, what is intron? 12th class topic. Okay. Now the next is not just enzyme, there are other molecules also which are able to regulate reactions or developments in the living system. So they are called as bioregulators. So how many bioregulators are there in the living system? It can be enzymes, also called as the biocatalyst. They can be vitamins, they even can be hormones. So all these three molecules, types of molecules, are able to control the rate of reactions and other things. That means they control the metabolism in the living system. So they are called as bioreactors. Now, like for example, I can give you an example. Functioning of the eyes is very important for us. It's a metabolic pathway which help us to understand what color we are seeing, or otherwise perception, which light, how bright is the light. So all this thing requires the retina to function properly, and for retina to function properly, rod cells and cone cells, few days back I covered in the protein topic, functions of protein. So that requires vitamin. So without vitamin, this is not possible. And in the same manner, I have told you, vitamins are part of the NAD and NADP, FAM, FMN and FAD. All these are important to control many of the reactions. That's why they are important as bioregulators.